Hello and welcome back to Jono's Lab. In today's video, I thought we'd explore a new release from Biohub. They're fully open sourcing a few different models for protein sequence modeling and folding and all sorts. And so I thought we'd talk a little bit about that space, and then I'd explore an idea that I had that I've been toying with the last couple of hours and I thought I should document. But mostly I just want to say, look, here's this cool new model and show some tricks that it can do and why that might be useful. Let me start up the code. So I'm going to connect to a GPU runtime and load the smallest of their model family, which is 300 million parameters, so we can start exploring that. And in the meantime, this is their protein atlas. So what they've done, they have a model that takes a sequence of amino acids, right? And I can actually show you in the thing. We'll have some sequence of amino acids coding for a protein, and this is going to fold up into some interesting structure. They have taken this model and trained it to predict if a particular amino acid was masked out, what that might be given the context. And so to do that prediction task well, it's learned all sorts of useful representations and features of the proteins. And so then they're able to look at those features and cluster together proteins that are similar. So that's pretty neat. They also are able to look at how the internal representations of the model fire for different features and highlights. You can see all of the amino acids in this region of the protein are strongly activating some particular feature, and these are from something called a sparse autoencoder, which is trying to say, okay, we see this sparse feature activating for these specific places. What do those have in common? They all tend to be rapidly bending alpha sheets, right? Alpha coils. Oh, alpha helices. Okay, what do these parts have? Oh, these tend to be terminal regions, or these tend to be trifoil folded structures. So they're able to start to piece together the roles of what some pieces of unknown proteins might be based on saying oh, this section of this protein that we're interested in, it activates similarly to this section in a different protein, which we know is involved in binding to such and such a molecule. So you get these really rich representations of these proteins, and you get this really powerful capability with this model for producing the structures, but also understanding what different parts of that protein might be. Anyway, so that's a lot of background to say very cool group, very cool models. Wow, it's doing a lot of requirements, I guess. That's no worries. I'm probably not going to run all the code again, but I thought it'd be nice to have at least the model loaded so we can experiment a little bit. So the stuff that I'd like to play with today is step one. This is a new model. I'm new to this field. I should caveat this whole video with that. But I want to just poke at it. I want to see what it looks like to put some inputs through the model. What do the outputs give us? What does that mean? What are some things we can start to investigate, explore from there? And then I also have in the back of my mind, I have an idea that this might be useful for generating variants of a particular protein. And I think that might be cool. Okay, so we're loading the model, and this is what it looks like. It's a transformer. It's a big stack of transformer blocks with attention and an MLP. And then at the end, there's the final language modeling head. Very vanilla. It's masked language modeling, so it's not predicting the next token. It's for a given position. It's predicting if that was a mask, what would that look like? So this code for loading the model and for putting a sequence through came straight from their README, right? We've loaded the model. We have it there. We have a sequence, which is in this case just the amino acid sequence for green fluorescent protein. And so our first step is to turn this sequence into some numerical inputs that we can feed to the model, right? And that's what the tokenizer does. It turns the sequence of amino acids into a sequence of tokens. Each one gets a unique ID. So you can see probably some special start token, one ID for each. If we compare the sequence with the inputs, there's two extra tokens, right? Probably start and end. So we can feed this whole sequence through the model, and we'll get the model output. And this is going to have various pieces attached, right? It's going to have a bunch of information that we could use. And so these are the logits, the hidden states, the last hidden state. And so let's think a little bit about what each of these is. The logits, there's 240 inputs in the sequence, right? 240 positions in this string of amino acids plus the 238 amino acids plus start and end. For each of those, we're predicting what the model thinks is the most likely amino acid at that position, right? So if we look at one particular position, let's say position 10, and we mask it out. So we replace the input with the special mask token, token 32. We can feed that through and see what the transformer predicts. Oops, I guess I missed some. Okay, we don't need that. Okay, sorry. I refactored this code a little bit. Okay, so we're taking in the sequence, the big one that we saw here, the sequence of numbers, which codes for this protein, except that in this position here, we're replacing the token there with the token 32, which is the special mask token. And so the original amino acid there was a G. And if we look at the model outputs, the logits is going to be the model's output.logits, and that's going to be the length of the sequence by the size of the vocab, right? So for every position in the sequence, we have 64 numbers. The higher the number, the more likely that that amino acid goes in that spot. So when we look at the top few, we see that the model actually thinks that this is the most likely amino acid, and glycine is down in second place. So it thinks there's maybe a 13% probability, then an 8%, then a 7%, then a 6%, then a 6.25%. So this is pretty useful, right? You can imagine saying, okay, what can this tell us about the sequence? If we find a protein, this model has been trained on billions and billions of proteins, and so it's learned some notion of what biologically plausible proteins look like. And so if you were to mask out an amino acid and say, okay, what's the most likely, the most plausible amino acid to sit there given all of the other amino acids in the chain, you can imagine that signal being a kind of strong hint as to what a likely replacement for that amino acid might be. And so a couple of cool things we can do. One is, what if we looked at the so-called surprise, right? Like how unlikely is the actual amino acid compared to what the model thinks should go there. And so you can see here, this is the some measure of that unlikelihood. And there's a few amino acids that stick out. It's like, hey, the model really didn't think that this one should go here. Now, one nuance that I'm going to digress into briefly, these masked language models are trained with some sort of masking ratio, right? So it's very rare that you'd have a 200 token input and you'd only mask a single token. And so instead, what you could do if you wanted an even more accurate readout of like what does the model think should go there, is you could do like a 10% masking ratio, and keep track for lots and lots of different random masks what the model predicted for each token in all of those cases and average that out. And so this is something that seemed like I could delegate it to AI, look many different runs with random masks for each position, keep running log. And it said, oh, look, glycine is now the most likely. But I didn't trust it, and I read the code. And sure enough, you'll see it's doing this, picking random mask locations, and then it's keeping track of the logits and accumulating them and then dividing them by the number of runs. This seems fine until you realize, like, a lot of the time we're feeding in the correct amino acid as the input. And in those cases, it's kind of obvious that the model should predict that same amino acid, right? So it only really makes sense to look at the output logits when the input token is masked. And so that's why the difference here, you see this is the sort of surprise graph generated by that first flawed attempt. This is the second time where I modified it to actually do what it should do. And now you can see the glycine is back in position 2. The probabilities have changed a little bit. So there's a slight difference in this versus the like single position masking approach. And actually, if you plot the two together, you'll see that they mostly line up. You could probably get away with just the single token masking. But for me, I'm happier to be closer to what the model was trained with, which is a higher masking ratio. And it also gives you kind of like some kind of almost regularization, or you're trying lots of different random maskings and then averaging. It's going to give you, for various variants of the sequence, what would generally be likely in this position. as opposed to like for this exact sequence with just this one masked, what does the model happen to predict is most likely? So to me, that seems like an even nicer measure. Cool. Okay, so that's one thing we can get out of this. For each token, we can get what are other tokens that might be a good, other amino acids that might be a good substitution in that position. We can also look at the hidden states of the model. So back when we were looking at the model outputs, it didn't just have the logits, it also had the hidden states from the final layer, as well as for all the intermediate layers. So this is 31, I think maybe input embeddings plus the output of each internal transformer block. And so we could look at these rich representations here. Each position gets a 960 dimensional representation out of each layer. And so that's pretty cool. That's what they use for some of these, well, not directly, but similar idea, right? To be able to annotate across a particular protein, like for each position, what features might that have? This would be one way that you could look and say, oh, especially here, it's activating this feature. So I thought, okay, well, let's look at the outputs and let's project them down using UMAP and see if similar amino acids tend to have similar representations. And sure enough, seems like there's definitely some clusters where they're all the same amino acid, right? Similar amino acids tend to have similar representation, but there's also some clusters that are quite mixed. And so then I also colored them by position in the sequence. And you can see here, this cluster here, they tend to have somewhat similar representations, at least in some dimensions, and they're all right at the start of the sequence. And so that's where you might start to get the idea of these all triggering some feature that's something like, a terminal binding section rather than the middle of some big globular clump. Okay, so that's starting to explore the different things that the model by default gives us. So far, we're not really doing much other than feeding some inputs through and looking at what the outputs are and starting to reason about what could this give us, right? This could give us interesting representations, which we could later cluster and bundle. It can also give us predictions for what amino acids are likely in a particular position. So then the next part of this is generative mutate. So this was the somewhat obvious once you've been playing around like this idea that I had to say, okay, it seems like one thing that people often do is you're trying to optimize a protein for some metric, right? And this can be a very slow process where you're tweaking things and then measuring in the lab temperature stability or binding affinity or how brightly it fluoresces or how fast its enzymatic activity is, right? You're doing some sort of physical assay, and then you could pick the candidates that do best and try and mutate them again. And you can do this with like actual physical mutagenesis, right? Like you're bombarding the DNA of the cells with ultraviolet light or something like that to try and shuffle things up. But that can be very error prone and slow, and you have very little control. Or you could be expensively just ordering DNA synthesis for each new variant you want to try, or some mix of both. But in either case, if you could cut down how many candidates you have to try in the physical world by generating more plausible candidates computationally, that would be incredibly useful. And so if you can predict even some sort of proxy metric that's going to be close to what you want, then you can start to optimize. And if you want to optimize, you could speed that up even further if you have a better mutation than random mutation. So that's a long-winded way of saying what I think might be kind of useful is to take this existing model. It's learned this idea of plausible substitution, right? Because it sees a mask token and it says, oh, I think it's either a glycine or a lysine in that position. Rather than randomly mutating a sequence, what if we used this model to inform our random replacements, right? And so that's all this is. So we have this generative mutate. It's going to take an input sequence. It's going to generate some number of mutated sequences. And for each of these, we're going to be picking random locations. And then we're going to be looking up in our list of like, what does the model think should go there? And we'll be using that to pick the token with some temperature. So it's like we're sampling replacements. And we can do this in batches. So anyway, rather than looking at the code, here's a nice visual representation, right? So here's a sequence, and we want to mutate this to get a few variants. We could just say pick a random location, replace that amino acid with another random amino acid. But instead what we're doing is we're saying pick a random location and then look at what's likely to go there. And then use those probabilities to inform how you randomly choose what goes there, right? So if it says there's a 50% chance that it's A, 20% chance that it's G, 10% chance that it's E, then we're going to say, okay, we'll roll the dice, but it's going to be a loaded dice. We're not going to just insert like methionine or something there. It's most likely going to be one of the top few predictions, right? So we get these nice variants of our initial sequence, but hopefully they're more biologically plausible and useful than the sort of random mutation if you just were literally picking one of the numbers with uniform probability. So, okay, let's try and actually put this into an example. This is all very, it's all very somewhat hand wavy. I picked a pair of chains that are in real life this cohesion Dockerin complex. It's related to like bacterial decomposition of cellulose and things like that. You have this big giant like structure onto which these useful enzymes are glued, right? So rather than having like just within your general internal part of the cell, you have these enzymes free floating around and you're trying to do stuff with them. This setup is to have a nice big, what do they call it? Scaffolding. That's the one I was forgetting. A number of these little cohesion chains bound together into some long big clump. And then the Dockerins are going to stick onto that. Anyway, not really important. Just was using AI to suggest some sort of likely candidates that we could start having fun with. But you can see here we want these to stick onto the scaffold and still do their job. So the ESM folks, I think they talked about releasing a full end-to-end like a small molecule binding workflow, and that might be an even better test. But for now, we're going to just be very hand wavy, right? We're just going to pick a scoring function, which is to do with whether or not the folding, because they do have the folding API, or you could run the model locally, whether or not it predicts those two chains that we'd like what their distance is, how closely bound they are, as a kind of very rough proxy for binding affinity, right? So we're setting up our situation whereby we're going to start with our candidate sequence, right? We want these two to bind together. And then we're going to start mutating both with random mutation and with our generative mutate function here, where instead of random replacements, we're using the sort of ESM model informed choices. And then we can compare on our sort of somewhat fudgy score function, we can compare those two and see how they do. And so here's the result that I got. The proxy score for the wild type, all of the random mutations were worse. Right, and this is pretty much par for the course with, I've seen this with quote unquote real protein engineering tasks too. Most random mutations are going to be a strict downgrade versus the existing like one that evolution has found. And this is unsurprising. There's such a huge space of proteins and most of them do absolutely nothing, right? It's very easy to generate garbage. But if we look at our modeling guided mutation, they're much less bad by our proxy score versus the random mutation. And in fact, one of them even beats the wild type, right? So I don't want you to read too much into this hacked together Colab notebook. But the core idea here, I think, is that using the model of what plausible biological proteins look like learned by this protein language model gives you a way to generate candidate mutations, which are less likely to just be terrible versus random mutation, right? And so then as long as you have some decent proxy function for, is this what I actually want? Like maybe you're properly modeling binding affinity or temperature stability, then this is perhaps a way to generate much more candidates really quickly that you could then try, and then you'd probably pick the top few of these that work in the simulation, and those could go onto the sort of lab validation or more rigorous, higher compute folding and so on, just to see whether that actually holds up. So that's that. Here's our modified, evolved, mutated candidates stuck somewhat close together. So anyway, that's all I wanted to show, this kind of exploration. I mostly wanted to, learn for myself what these models look like, what they can do, start getting some ideas of what else we could try with this. You definitely can run at least some parts of this downloading the models from Hugging Face rather than using the API. For this small test, I just did one set of mutations and quickly fired it off to the API. But we did use the model locally for generating those mutations. Yeah, let me know if I've made any egregious errors or if you have any questions. I'm going to keep playing with this, but I thought while it's all still fresh and I've been having fun, might as well share it with all of you. I'd encourage you to go check out the atlas. It's really fun to explore, and if you've got like if you've got a protein in mind, you can chat with it and explore, just seeing the nearby proteins and seeing if any of them look like they might be similar, or I don't know. It's very inspiring to be able to look around and dig into the different clusters, and then to zoom out into the wider context and see, wow, there's this whole universe of form and function of these amazing folded squiggles that drive all of biology. So I don't know, I find it really inspiring. I hope you do too. Thank you so much for watching, and we'll see you in the next one.